one thing that's very clear, and again, I'm surprised it's not a bigger chunk of sort of discussions around innovation in biopharma innovation, right? Is that how important IP is in biopharma innovation, and it's almost like people sort of think of it as a bit like the sort of air they breathe. They don't really talk about it. You're so you're sort of swimming in it, so you don't really notice.
Today, I'm speaking with Jack Scannell. Jack studies biomedical R&D productivity from both an economic and a scientific perspective, particularly the unpalatable contrast between cheaper and better scientific inputs and what has been a long-term decline in innovative output efficiency. He co-founded Atheros Pharmaceuticals, a company that works on a new small molecule drug class that extends mammalian lifespan and neural health span. Jack and I discussed the politics of measurement, the entanglement of science, money, PR, and biopharma, the changing potential for serendipity in R&D, as well as his 2012 paper that coined the term Eroom's law for decreasing R&D efficiency in biopharma, and what has changed since the time of this article. Without further ado, I hope you enjoy our conversation. So, Jack, thanks so much for being here today.
Well, but it's a real pleasure. Thanks for inviting me, or I should say re-inviting me. Although it was so long ago that I did it, I've forgotten what I've said last time. So I hope I don't contradict myself. So anyway, there we are.
Here we go. So to start with, you've been asked about your Eroom's law paper for more than a decade now. And while I will have some questions about it too, I thought it might be interesting to start with asking you about what you feel like is under-discussed in the R&D productivity context. What do you really like to talk about that usually doesn't get the time of day in terms of what people have asked you?
I still think, actually, this is going to sound boringly like Eroom's law. I still think that the quality of diagnosis of the R&D productivity problem is poor. Right, so this is a really important it's a sort of really important matter of public policy. You've got a situation at the moment where I think on the best measures, I mean, you can argue, you can quibble about the measures, but on sensible measures, it looks like private sector returns on biopharmaceutical R&D in aggregate are below the cost of capital, right? Nonetheless, society does want good new medicines, right? And although I think, hope springs eternal in biotech entrepreneurs and drug company chief executives, and they don't like to admit the fact the financial returns are what they are on average because they all think they're better than average. It's not a good situation for the world to be where it's so difficult to sort of generate, positive, where people are actually losing money by doing R&D. And I think almost by definition, because people who are actually actively engaged in R&D don't find that a particularly palatable topic, I think the causes are not well debated in public. And I'll give you an analogy here. There's a wonderful British political satire, which isn't on TV anymore, but used to be, called Yes Minister or Yes Prime Minister. And there's a sort of logical fallacy which the civil servants who always take the mickey out of the politicians joke about, which is the politician's fallacy is this. It's okay, things are terrible, something must be done. This is something, therefore we must do this. So typically what happens is, rather than diagnosis, what you get is people arguing for their pet project to be the solution to the problem.
So you talk a lot about kind of jumping off of the misdiagnosis or the difficulty still of diagnosing what exactly causes declining R&D productivity. I guess this is a good point to maybe, you kind of talk about two main factors 12 years ago in that paper, but what have we learned since then about what might be other causes that are leading to declines in R&D productivity? Or maybe even like what has changed in terms of the methodology with which we can, like the ways of measurement with which we can kind of like determine potential causes.
Yes, I think good measurement is difficult, and I'm actually not convinced there's a huge amount of progress being made in terms of measurement. So I wrote a chapter for an OECD book, I think last year or the year before, and the book was about kind of AI transforming innovation efficiency, right? And I didn't write about anything as interesting as that. I wrote about measurement problems, right, when it comes to R&D productivity. The useful measures of pharma R&D productivity are quite hard to make, right? So there's things that are easy to measure, like number of new drugs per $1 billion spent. But we know that the value of new drugs is terribly skewed. So on one hand, you've got kind of COVID vaccines that, revolutionized the world and let people get back to normal lives quite quickly. And then on the other hand, you've got certain new drugs that really don't do very much at all, right? And so not all drugs are the same, and so some approvals are hugely important, some aren't. And similarly, the sort of financial returns on individual drugs are very heavily skewed as well. And arguably, the things that society is really interested in, which is sort of number of major human welfare gains, they're even harder to measure. So I think there are real measurement problems. I think that, the financial measures are pretty easy. Not that everyone does them right, but it's pretty easy to sort of measure financial performance. But I think there's a problem, which is the things that we'd really like to measure are really hard to measure. And because of that, the people who do measure them often skew the measures depending on what they want to find, right? So, there's people who don't like the drug industry who argue that actually all new drugs are rubbish and none of them have any, value at all, et cetera, et cetera. And then, of course, you have other work, sometimes sponsored by the drug industry, that says, it's incredibly valuable. But lots of the debate is but what people are really having, they're sort of kind of having a debate about measurement methods, and they're dressing up as a substantive debate, right? So you could pick ways of measuring drug R&D that don't sound preposterous, that give you rather different answers, right? So it's very difficult. And, without wishing to digress too much, I don't think this is going to happen, but I think the world would be better served if there was a bit more sort of attempt to come up with some sensible consensus on measuring the value of biomedical innovation. Because at the moment, it's easy to have kind of rather meaningless policy debates because people can't agree on, so it's very hard to know how much we should pay for new drugs, right? If there's no consensus on what the value of new drugs is, right? And I think that's the sort of situation we're in.
So in that vein, are people working? Because I feel like, it is an incentive problem in terms of like the drug industry perhaps wants to get to very different conclusions and will choose their measures accordingly than, say, like, anti-drug industry groups. Like, whose job is it to actually think about standards that are taking into account the welfare of society, say,
right.
Which obviously is like a troubled
So I think I may be being unrealistic
here.
here, right? And this is sort of shows a kind of sort of technical, geeky bias. But one analogy I would point to is something called the Stern Review, which is maybe even 2 decades old now, certainly more than a decade old. So some time ago, the UK government paid a very eminent economist to look at to try and take an economic view of climate change. Right? And I'm sure lots of people would disagree with Stern, but I think the Stern Review was a serious attempt to try and at least produce a sort of a consensus that maybe the sort of middle 50 or 60% of people could agree on, right? And I don't imagine, there'll be 20 or 30, there'll be 20% of people at each end who won't agree. But when people ask me about that, I say, well, the best model I've got of people doing this is the Stern Review, which, the kind of biopharma industry needs a kind of Stern Review, where at least the sort of middle of the sort of political world can agree on roughly, some of these things, and also agree on some methodologies, right?
So maybe getting more into particular aspects that you talk about in your paper, I wanted to ask a question about what I might, term the Chesterton's fences of R&D. As an example, you talk about the danger of reducing R&D spending without understanding the factors that are affecting R&D return on investment. Have you encountered other examples of these Chesterton fences of sorts, like just things that might be in place, mechanisms that might be in place, or roles that might be in place in a given organization or industry more broadly, that maybe their rationale isn't understood. Maybe their history of like becoming established in the industry isn't really thematized. But if they're taken away, that might still be pretty dangerous in terms of like having unintended
Could you
consequences.
give me a couple more concrete examples? So I haven't come across the term Chesterton's fences, so I feel
Oh,
I'll
okay.
give you a really
So
bad
yeah,
answer if I guess what it means.
no, absolutely. So this is a concept that was derived by the philosopher, among other things, Chesterton, who talked about um, only removing a fence that was standing on a hillside that you maybe like you're coming into a new territory, you don't really know the ways of the land or the people or anything like that. Basically, you're like the naive outsider that's like, why is this fence standing there? It's obviously not protecting anything. There's no cattle, no nothing. Let's take it down. And actually, there is a reason why it's there. You just don't know that reason yet, and it might be like not uh, legitimized in like a I don't know in the way of thinking that you come to the terrain with, but it still has a reason of being. So I
Sort of
wonder what
they're
the
things that
equivalent
appear
would
to be
be.
barriers, but if you understood the system better, you would realize they serve a function that you hadn't previously guessed, right? Um,
Precisely, yeah, much more succinct.
um, I mean, I think the most obvious ones, or rather, if I think about where my view of sort of barriers have probably changed, right? So things that I thought were sort of unreasonable barriers, and now now I don't. Um, I don't want to sort of paint drug regulation as perfect, but I think over the years I've got more sympathetic to. Um, I've become more sympathetic to the idea that actually many things around drug regulation are perfectly reasonable, right? Or rather, that there are trade-offs, and the idea that sort of regulators produce sort of unreasonable safety or efficacy standards, and innovation would be so much more easy if we could do things that we did in the 50s and 60s. Well, I think probably as I've become a bit less ignorant, I think I've come to the view that a lot of what we did in the 50s and 60s is probably quite a good job, right? It's really quite a good job. It can't be done now. But it's interesting. I haven't really thought, but the question is an interesting one, right? I think, rather, I do think there is something about it though, because I think one thing that I'm struck by is, I'm old enough to know that there've been a number of generations of people who've come from outside of the drug industry, looked at it, thought it looked preposterous, decided they could do much better, and then promptly didn't do much better. Right. So for example, tech entrepreneurs have been rescuing the drug industry for quite a long time now, right? And it still ain't been rescued. Now, that doesn't mean they never will, but it does imply that there was something a bit like the Chesterton's fence phenomena. And, lots of people have looked at this, and said, oh boy, we can do so much better with, coming from the industrial background we've come from. But actually, they haven't done much in aggregate. They haven't done much better.
I think that I gleaned a little bit of that in your paper too, where you're kind of talking about physicists, and also I think you're talking specifically about tech entrepreneurs and just people from the semiconductor industry too, where solid state physics being uh, slightly more predictable and slightly less complex than biology, um, kind of come with the same metaphors and frameworks that maybe worked in that sector, and try to apply them to drug development to an extent that just isn't reasonable for the
And of course, it
system that
doesn't
you're
mean that
dealing
it
with.
never will, or there aren't things
Yeah,
to learn, right? But I think it's easy to underestimate how difficult it is, right? Uh, uh, if you haven't tried it, um, and I've tried it a couple of times. I worked for a little biotech company a number of years ago, and I'm running a little biotech company now. And it ain't easy.
in terms of the biological metaphors, I found really interesting in your paper, and I have some more questions about kind of the context from which you come. As I understand, like you did a PhD at Oxford in computational neuroscience, and went on to industry in various roles afterwards. But I find that there is a focus in your writing on, even your Twitter bio says like, it's like drug development, money, science. Those things are intertwined and mutually working on each other, influencing each other. So I'm really interested in how you think monetary incentives have shaped biological metaphors in the last few decades. The basic research brute force bias, for example, in the Eroom's Law paper, kind of match the inclinations of many commercial managers, management consultants, investors, and such. Like, how do the biases and the temperaments and the incentives from different people, maybe outside of the science, still like shape the way that biological research has progressed?
I mean, there may be several, but I think one quite powerful issue is, and I don't know if this is just money, but I think it's partly money. Survivor bias gets mistaken for prediction, and then people industrialize the stories that are very heavily contaminated by survivor bias, right? So, for example, not for example, to be concrete, right? Every single drug that works, pretty much, with few exceptions, has a story with it that sort of retrospectively makes sense and makes the drug sound very rational. I'll give you an example. There's a drug called Avastin, bevacizumab, big selling anti-cancer drug. And there was a sort of rational, well, at least there's a sort of story that came with bevacizumab when it was first launched, which I've no particular reason to, well, it was a story that most people who knew about the compound told about it, which was that, cancers can only grow if they grow a blood supply as they grow, right? Because otherwise, if you don't grow a blood supply as a tumor, then your growth is very limited, right? So what you need to do is you need to give this agent which stops vascular endothelial growth factor having its effect, and therefore you stop tumors growing new vasculature, and therefore tumors will stop when they reach a certain size. They won't kill the patient. And by the time I stopped looking at bevacizumab or Avastin, the story had changed a bit. And in fact, some people were arguing that it was doing vascular normalization. So it wasn't stopping tumors growing new blood vessels. It was letting them grow normal blood vessels. And then that way, when you gave chemotherapy, it killed the tumor. Another example is lithium, is a compound that was discovered serendipitously in bipolar disorder. And I remember, during my medical training and then subsequent academic career, the story about how lithium worked completely changed. Gleevec, another good example, which was initially sold as being very sort of ultra targeted, and then when it started doing a broader range of things involving other receptors, people wrote about it as a sort of broad spectrum, as a sort of broad agent, right? But the issue here is, at every step of an R&D process, you have to have quite a clearly articulated story about why the drug is going to do what it's going to do to convince people to give you the money to take it to the next part of the process. And of course, most things don't work for reasons on which the mechanistic story you had at the time was entirely silent, right? But you're left with things that emerge from this process with a story, right? But the story may not have been predictive and might not predict why those things ever succeeded, but they all emerge with a story. And then I think there's a tendency to confuse those stories with prediction. So you sort of industrialize a process that sounds like those stories, right? So the sort of analogy is sort of Darwinism versus intelligent design, right? So, creationists look at the eye and they say, look, this thing could not possibly have just be the result of sort of selection acting on random variation. Someone must have designed this, right? And I think there's an element of that in people looking at drugs, right? Now, clearly they are designed to an extent, but I think we underestimate the extent to which there's a selection process, which and things are selected on properties we don't really understand or didn't predict, right? Or they fail for reasons that we didn't predict. And you could imagine reconfiguring drug R&D slightly differently if you actually paid more attention to the role of serendipity, right? You would, for example, search for longer. You would maybe during sort of late preclinical, early clinical development, you would try and keep more opportunities open for longer, right? You wouldn't rush to have an individual particular target product profile quite as quickly, right? You would realize this is the sort of mapping from drug to disease is sort of one to many, not kind of one to one. And yeah, you would have a sort of broader search strategy, not such a kind of narrow strategy, right? So that's kind of one example that strikes me.
Yeah, the role of serendipity is another thing that I wanted to discuss. It does seem like, from even your paper and just others that have commented like in the show and beyond on kind of modern R&D, design, it seems like the, I don't know, surface area on which serendipity can dock on is rather contracting than like expanding. And I wonder whether you have a sense of when that uh, shift or that trend toward more and more maybe like, I don't know, whether it would be fair to say Taylorist, like, R&D management, but like very rational R&D management and away from like keeping open enough kind of space for serendipity to emerge. Like when did that arrive?
So first of all, I'd emphasize what you said about sort of serendipity. And I think so this is not a minor feature of the economics of the drug industry, right? So it's if you look at arguably the two best-selling drug classes in the world at the moment, which are the anti-TNFs and probably the PD-1, PD-L1 agents, right? In both cases, there were huge fat chunks of sort of serendipity and sort of bare survival of these agents, right? So, and I may be misremembering the PD-1s, PD-L1s, but I think they came out of attempts to develop adjuvants for vaccines, right? And in fact, it may be one of them was meant to be an agonist, and actually ended up being an antagonist instead. Sorry, an antagonist and being an agonist instead, right? So, and then similarly, the anti-TNFs were initially tried as septic shock agents, right, before a guy called Mark Feldman and others realized that they might have a role in autoimmune disease and rheumatoid arthritis, right? And actually, the list of very important drugs with histories a bit like that is long. It's not unusual. It's very usual. So, so sort of drugs have a kind of biological mechanism, and people are not very good predicting initially what that biological mechanism might be useful for. I do think that if you go back as far as the sort of 50s and 60s, and I may be being a bit biased here because I think I know the history of psychopharmacology a bit better than, not a bit better, probably better than most other therapy areas. The nature of R&D was such that compounds were tried in people relatively early. So effectively you had something that kind of resembled phenotypic screening in humans. And arguably, although that would raise a whole bunch of sort of ethical questions if you did it now, it was a search strategy that actually made those things that are now regarded as now called serendipity much more likely to happen. So for example, the first antidepressant was, it wasn't really synthesized with the view that it was going to be an antidepressant. And indeed, until antidepressants came along, the sort of diagnosis of depression hadn't really been crystallized in the way that it is now, right? So antidepressants, to a certain extent, created the modern diagnosis of depression. But these sort of psychoactive compounds were created, right? And people thought, well, it may be a sedative, it may help in patients with schizophrenia, let's give it to different sorts of, and then physicians and also nursing staff observed what happened. And I certainly know psychopharmacologists who argue that there is not one single class of antidepressant that was discovered in a lab. They were all discovered by phenotypic screening or observations in humans, right? And I do think the sort of, having said I think my concern about regulation may have been an example of a Chesterton's hurdle. Right now, I'm almost arguing the opposite point, where in certain classes, right, you sort of engineer serendipity out of the system by effectively forcing yourself down a very sort of narrow, long path before you try a compound in people for one very, very specific niche indication.
That's a really interesting point regarding how having a solution that serendipitously arose, like antidepressants, actually has an effect on the way that we diagnose in the first place.
Yeah.
Like
So
those
the
two
first
categories being like
the first
mutually
class of
reinforcing.
antidepressants were effectively sort of, if I recall correctly, kind of antihistamine derivative that a guy tried in a inpatient population in patients with, what would at the time was called schizophrenia. Well, what is now schizophrenia, and I think it was probably called schizophrenia at the time, although psychiatric diagnoses have changed over time. The second class of antidepressants was effectively a failed anti-tuberculosis compound, where it was given to, again, patients with tuberculosis who were pretty miserable because they had tuberculosis. And they noticed that the patients cheered up a lot, their moods improved, but their tuberculosis didn't get any better. Right. So in psychopharmacology, it's happened a lot of times.
I also wonder, I mean, the kind of monetary aspect of biology obviously hanging over everything that we've been talking about so far, and also just your work more broadly. um, when did you start to think economically, or more in monetary terms, within your own career, about biology and like what shapes it?
So this might sound sort of weird, but I think when I was an undergraduate, I studied sort of medicine, but I also studied zoology. And a very interesting bit of zoology is behavioral ecology. And behavioral ecology it's a bit like sort of animal economics, right? And it's quite interesting. I found that the sort of behavioral ecology and a lot of it's about sort of optimization, cost benefit. What's the risk of pursuing food source A rather than food source B? How do you, as a foraging ant, optimize the amount of, palatable grubs you can collect in a bit of desert? So I found when I started working in business or commerce, funnily enough, it a lot of stuff sort of chimed a lot with sort of behavioral ecology thinking. And this isn't meant to be a sort of non-answer, but it's sort of, in a sense, I don't think there was ever a sort of aha moment. It just always seemed to me obvious that a lot of behavior is driven, but particularly, a lot of behavior by companies is driven by money, right? That doesn't seem to be a very controversial. I shouldn't think that shouldn't be a very controversial view. And I think there's lots of really obvious things, right? So for example, one thing that's very clear, and again, I'm surprised it's not a bigger chunk of sort of discussions around innovation in biopharma innovation, right? Is that, how important IP is in biopharma innovation. And it's almost like people sort of think of it as a bit like the sort of air they breathe. They don't really talk about it. You're sort of swimming in it, so you don't really notice. But, why do we have a drug industry? We have a drug industry, not entirely, but largely. Or why is the drug industry so big? It's big because novel chemistry has particularly sort of robust IP associated with it, in the way that, for example, a novel talking cure or a novel medical device may not, right? It's easier. I think it's kind of often easy. It's harder to sort of make So there's something around the sort of patentability of novel chemistry that means lots of capital flows into drug R&D. There's no particular reason to think novel chemistry is the best way of deploying innovation capital for human health, right? But it's the thing that sucks in most of the innovation capital for human health because it's the thing that's most appropriable. And I think you can see a huge amount of sort of biases in pharma versus other sort of possible flavors of medical innovation. And even within pharma, you've got something, the minute, old compounds, people suddenly lose interest, not because you couldn't do something useful with them, but because if you can't appropriate the IP, it's very hard to raise the private sector capital to do stuff with them.
Yeah, and funnily enough, Paul Krugman, I think, was the one that wrote an entire essay or gave an entire speech on what economists can learn from. He was focused on evolutionary biologists,
Okay.
but
Okay.
He actually, I found that 2 years ago and was really interested.
And also, setting aside IP, there's other funny things like that, sort of historical accidents in drug pricing, right? So why is it that roughly half of all the drug industry R&D at the moment is going into oncology? Right, it's because you've got a kind of oncology pricing bubble in the US where you have a combination of state and federal legislation that means cancer drugs have to be reimbursed. It's because you've got ASP plus, selling price plus markup reimbursement, and it's because lots of people who prescribe cancer drugs make more money if they prescribe more cancer drugs. So it's nothing to do with medical need. Well, I mean, clearly oncology is a major unmet medical need, but you have some, but then so a lot of neglected tropical diseases, right? And it's, so if you want to understand why people do what they do, it's not just about following the money, but following the money is important.
Yeah, 100%. Yeah, and I think just a general awareness of the contingency of our notions or like our research tracks is rather useful, but I wonder actually how much you encounter that kind of in the people that you work with too. Do you feel like there's a general, I don't even know what category of thinking to put this in necessarily, but it seems like you're thinking very philosophically about biology. Like, what are the frameworks, the metaphors that we think about? How are they contingent? What are the histories of them? Are they rigorous now? Do you feel like you developed that in academia or like beyond that once you went into industry? And did you feel like that's actually a common approach to think about, especially the world of drug development?
So it's funny. I think at least in one chunk, not all, but in one chunk of industry in which I worked, and I don't know whether this says anything about me or about the environments, right? But I worked doing drug and biotech investment for a company called Sanford Bernstein, which is arguably quite a scholarly company, right? But actually, I found that the sort of thought processes in working at Sanford Bernstein around drug and biotech investment were actually very, very similar. There didn't seem to be a huge, massive difference between that and some of the stuff I was doing as an academic, right? The subject matter was different, but there you were sort of, paid to think about stuff, and indeed some of the sort of time trade-offs were similar. So in sort of again in financial markets, you want to know things that other people don't, or you want to be able to sort of predict the direction of change of other people's minds, right? And in some ways, at least in bits of academia, there's a sort of similar, and also I think the idealized rigor was similar, right? So not that one is always successful, right? Plenty of stuff you do is kind of quick and dirty and not very good. But my sort of first foray into investment, it was a place where they were very happy to let you think about things for a while and chew them over before you had to necessarily talk to anyone about it, right? It was sort of quite thought oriented, not necessarily, so think before you open your mouth or write stuff. So actually I maybe didn't find as big a difference as you might expect, and I can imagine a lot of my Sanford Bernstein colleagues would have probably been happy academics, right?
Interesting. There is another question I tend to just like to ask, and you can apply this to the work that you're doing now. So I suppose you're leading a small, you said, biopharma company yourself right now. What's the implicit knowledge in that field that you feel like holds true generally, but it's not necessarily explicitly stated? In other words, what has to be learned anew by everyone that's entering the field with no real good reason? It could be written down, shared more publicly. Like, what do you feel like you kind of had to learn by running into walls, and maybe unnecessarily so, as far as you can tell, anyway?
So just so I understand the question, is it almost like, what is it, if it was systematized and written down, would make life easier for everyone who's in my position? Is that the question?
Yes, yeah, yeah. Just kind of, yeah, bits of knowledge, just understandings that maybe senior people have learned the hard way,
Yeah.
but it's kind of established itself that that's the way things are. But uh, maybe it's like a Chesterton's fence of another sort
Yeah.
too. Like
So
there is
I
the danger of
think
just thinking it could be, but
so I think I'm sure lots of little biotechs have this actually, but it may be harder to solve than I imagine. Right. So I think it's probably fair to say that so five of us founded this biotech firm, and I think it's probably fair to say that I'm the baby of the team, right? So um, it's a pretty experienced team, and uh, so five of us founded the firm. I'd say there's roughly sort of two and a half of us, 2.5 FTEs or 3 FTEs, right? So I work pretty much full time on it. There's a chemist works full time. We've got one of the scientific founders who's sort of notional half time, and then the others are more sort of, I'd say, sort of board type roles or specific stuff when we need it. But what you find is most people, I guess, doing what we're doing will have areas where actually they are quite expert, or and they may have more than one, right? So unsurprisingly, our main scientific founder, Laura Duggan, knows an awful lot about the compounds that we're working on and their properties in a whole bunch of model systems, right? She knows how to make. Subhashish, our chemist, knows how to make these compounds, which not many people know how to make because they're very peculiar. But these are sort of islands of profound expertise in a very complicated process where actually there are bits around which you know surprisingly little. So I sometimes say this when I'm sort of trying to get CROs to do work. I'll say, I kind of know the words that describe what you do, right? So I can sort of say it, but actually my knowledge of that thing is completely superficial, right? So other than knowing there's a word that describes what you do, I don't actually know what what is underneath that word. But there will be people who've spent 30 years doing that and still think they don't know it, right? So I think the tricky thing, I think, is it's fitting, it's understanding the sort of broader process in a sensible, logical way, and trying to sort of navigate this much bigger process when actually the people in a small team in a biotech will probably have narrow expertise, but you haven't had to put it all together. Now, the reason I think that maybe not as easy to fix as you think is because it may be that the manual on how you build a biotech company is so long and complicated that actually, you'd have retired before you got to read it all, right? So I think there's a sort of tricky thing, which is what would the sort of biotech founders manual look like, right? And in a sense, you're stuck between it being sort of trivial. You say, well, I know that already, or being so long and detailed that you say, that so I think that for me the tricky thing, or the thing that would be brilliant if someone could write it in a way that was short enough to read, right, in less than a month or 2 months, it would be the sort of how the bits fit together, and sort of very detailed. So it's almost kind of detailed checklists, right? And sort of, this is the order you do things, and these are the things you've got to do, because again, that is assembling that jigsaw puzzle is tricky if you haven't done it before.
yeah, yeah. And I guess I mean, it always depends on like, how high of a level you're analyzing these things from. Like probably from high enough, with broad enough strokes, a lot of the process of starting a biopharma company looks rather similar. But then like how much information that actually is like the granular stuff that you want, that you
Yeah, and
maybe
then you'll
don't
run
know
into
yet.
some
It's
specific
non-trivial. Like,
problem, like
yeah, yeah.
that that you run into some sort of specific, kind of weird manufacturing question that, so for example, nothing terribly problematic or bad, but we're trying to make a class of compound that no one's ever made before, or and the chemistry is kind of tricky but not impossible. But you've then got this whole set of stuff around. Okay, well, how if you've got some new chemistry, then that then presents a whole bunch of sort of new questions that then takes quite a lot of time to sort of takes quite a lot of time to solve. So, but again, I think for me the tricky thing is it's sort of placing the islands of knowledge within the sort of broader process, and then really trying to understand what you need to do and when, and at each point where to allocate your limited time and resource, right? Because clearly there's a whole bunch of questions you've got to answer before you start human trials, right? But actually there are cleverer and there are sort of more effective and less effective ways of ordering that sequence, particularly if you're trying to coordinate it with fundraising, right? Because there's certain things that are expensive, and so what were the things you would do to create maximum confidence now to make it easy to raise money? So that's requires a lot of thought and is tricky.
Yeah. I know our time is running out, but can you think of any important questions that I have not asked you today? Things that you really wanted to address that have been left undiscussed as of yet?
So let me see. I mean, there is my sort of pet enthusiasm about models, right? Which you may or may not have read. I don't know. And I'd be happy to sort of answer another question on that if you want to ask one. But it may be I think I may have just talked a lot about that in the last podcast, actually. So it may simply be rehashing old ground. So I'm actually happy not to be asked about it if you think it's in there already.
Yeah. Well, in terms of models, are you talking about especially the Eroom's Law paper and kind of the follow-up to that?
No, so there's a paper in 2016 and then one in 2022. So I think in the Eroom's Law paper, I may not have said it in quite this way, but I think I may have said it in this way actually on reflection. It's that if you've got a process where all of the inputs seem to be getting hundreds, thousands, millions, or billions of times cheaper, but the process has got 100 times more expensive between 1950 and 2010, then there's two broad classes of explanation, right? One is you've run out of some critical resource. So actually, maybe R&D really is getting much better, but actually we've run out of things to find, right? That's one sort of logical class of explanation. And then there's another logical class of explanation, which is actually what you've done is in making things more efficient, you've also introduced qualitative changes. So you're actually doing things differently, and you've sort of accidentally industrialized the wrong set of activities, right? So it superficially looks like so you've reduced the unit cost of activities that are actually fundamentally not very productive, right? And stopped doing things that might have had high unit costs but were more productive. And those, of course, are not mutually exclusive, right? And I think it became clearer to me after the 2012 paper that if you both look at history and also do a bunch of sort of geeky decision theoretic modeling where you regard R&D as a search process and try and understand the parameters to which search efficiency is sensitive, what you find is that the ability of screening and disease models to accurately predict human outcomes seems to be extremely important. And this provides something of an explanation for Eroom's law, which is the screening and disease models that give us the right answers. So, for example, a lot of anti-infective models are pretty good. Anti-hypertensive models are quite good. The models for stomach ulcer or stomach acid secretion are really good. Those give us lots of effective drugs, which ironically render the models commercially redundant, right? And what happens then is you're left with the models that don't identify good drugs. So ironically, because they don't identify good drugs, they remain in use for decades, right? Because they don't render themselves redundant. And there was also some sort of more quantitative work that showed very, very small improvements in the quality of models can actually have quite a big impact on your your ability to identify good therapeutic candidates. So, that's something that I'm sort of still very interested in. I think it's correct, but it's a tricky problem, not least because the financial incentives don't really stack up, right? So you get rewarded for novel chemistry, but if you have a better model that identifies a new therapeutic mechanism, then actually a lot of that information can leak to other people, right? So you don't really have you have inadequate private sector incentives for improving the quality of models. Now it may be there are ways of solving that problem, right? And I know that lots of the sort of new hot, I would say, companies at the intersection between sort of AI and biology are trying to address that problem, right? So building models that they can characterize that they think are better. But nonetheless, I think sort of where I've come to from the Eroom's law is it's both a resource depletion problem and a kind of misindustrialization problem. But the trouble is, it's the resource depletion has kind of has forced the misindustrialization, right?
Yeah. Interesting. Yeah. We'll make sure to link to that paper, and as well the Eroom's one as well. Thank you so much for taking time
Not at all.
today. I really enjoyed this conversation.